Hello, my dear students. Welcome back to the channel, and we continue our journey toward academic excellence. And today we are going to discuss another topic, that is human nutrition, and it is also known as animal nutrition. This topic is for the students of O levels, IGCSE or GCSE. Whether you are appearing through Cambridge, Edexcel, AQA or OCR. So this topic or this video lecture will definitely help you people. In this topic, we are going to discuss about food and the different components of food, and we are going to discuss about digestive system and its functioning. When it comes to food, what we eat as breakfast in the morning, as lunch in the afternoon, or as dinner in the evening. So what is that food? That food is actually mixture of different nutrients. So now the question arises, what is a nutrient? A nutrient is a substance, a chemical substance that is a part of your food, and this chemical substance provides you with energy, and this substance provides you with the raw material. With the energy and with the raw material, you can perform life processes within your body. Your cells can carry on the life processes. Means these nutrients are providing you with the energy and with the raw material that can be used within the body to raise new cells, to maintain growth, to maintain healing and repairing process, to develop immunity. And the energy is used for movement of the body organs, for the movement of the individual as a whole, or even for reproduction. So nutrients, there are a number of different types of nutrients. And why they are of different types? Because a single nutrient cannot meet all the requirements. Means different nutrients have got their different significances. And that's why we need multiple different type of nutrients in our food in different proportions, so that these nutrients in the mixture in the form of a food can meet all our requirements. Generally, we classify nutrients in two categories: micronutrients and macronutrients. And this segregation is done on the basis of their bulk requirement, and this is done on the basis of their significance in the life. Micronutrients are the ones that are needed in very minute quantities, in very small quantities. These nutrients are likely to support various essential functions of the body. These nutrients, if they are not present in our diet, or they are in less quantities in our diet, so we can still manage our survival, but with miseries. But we can still maintain our survival. Like minerals, like vitamins. So these are present in our food, not in the bulk amount, but in very minute quantities. And we can have various life benefits through these minerals and vitamins. But when it comes to macronutrients, these are the nutrients. We cannot survive without these nutrients, because these nutrients are going to make structural components of the various cell components, various cell organelles, like carbohydrates, fats, and proteins. That's why these nutrients are present in larger amounts, in larger masses in our food. And these nutrients, like fats and proteins, they are required to make membranes, including cell membrane. Carbohydrates like glucose is needed for respiration and generate energy. Similarly, proteins are needed to make enzymes that are to catalyze, that are to speed up the chemical reactions that occur inside the cells or within the body. So without these nutrients, our survival is not possible. So they are very integral component of our diet. And that's why the maximum mass of our food is contributed by these macronutrients. Dietary fiber and water, they have also got their essential importance, and we will be discussing them in the category of macronutrients. First of all, we proceed with the micronutrients, and we discuss minerals first. Minerals are very important to us because these minerals, when they are serving as component of various enzymes, they are making those enzymes to work efficiently and effectively. And certain life molecules, they also have these minerals as essential components. For example, without calcium, bones would not be existing. Without iron, there is no concept of hemoglobin. Without magnesium, there is no concept of chlorophyll. So these minerals are very important because these minerals help enzymes to work efficiently and effectively, and the chemical reactions proceed at the required rate. And these minerals help us to build essential molecules within the body for essential life processes. Over here, we will be discussing two important minerals. One is iron and the other is calcium. Why do we need calcium? Calcium is very important because our bones, our teeth, they have got calcium phosphate, a calcium salt, a very essential component of our bones. This calcium is not only required for bones. This calcium is required for healthy nervous communication. This calcium is required for healthy muscular contraction. And this calcium is also important when it comes to memory because the brain cells, that is the nerve cells, they maintain their communication with each other. So that would not be possible without calcium. So this calcium is very important to us. And if we do not have enough calcium in our diet or even no calcium in our diet, so that is going to create a lot of problems to us. So that is what we call deficiency disease. The main deficiency disease that occurs because of calcium deficiency, this is rickets. And ricket is characterized by weak, soft bones that are likely to deshape or fracture even with a smaller pressure. And why is it so? Because there is no calcium to keep these bones strong, to keep these bones thick. So when calcium is not there, the bones become thin, they become fragile. So they can easily get D-shaped or fractured with even a little pressure. This calcium deficiency also results poor nervous communication, poor muscular activities or muscular contraction. Again, the people might be facing headache or may not be work very efficiently, any muscular activity at all. But where do we get this calcium from? So calcium can be availed mainly from milk because milk and its characteristic color is because of calcium dissolved in it. So milk is an essential source of calcium and it is readily available to everyone in the world. Other than milk, calcium can be obtained from the food sources, from the food that we can obtain from the sea, from the oceans, like fish, prawns, shrimps, lobsters, etc. So they are also a rich source of calcium. Certain vegetables or fruits can also provide you with calcium, but with a little amount. Iron is another important mineral, and iron is needed for making hemoglobin, a protein found in the red blood cells that is responsible for the oxygen transport. This iron is needed for making hemoglobin, for making red blood cells, and if iron is deficient in our diet, so definitely hemoglobin formation will be affected. Less hemoglobin will be formed. It means less red blood cells will be formed, and less red blood cell means anemia, a disease where oxygen supply is reduced to the body tissues, body muscles. And in that case, body tissues, body muscles are not able to maintain sufficient aerobic respiration to meet their energy requirement. So in that case, they would not be able to serve, do any muscular activity effectively. They would not be able to work. They would not be able to memorize anything effectively. So if we want to avoid this deficiency disease, anemia, we need iron. And where we can have this iron from? So iron we can obtain from green vegetables like spinach. We can obtain iron from fruits, apple, banana. They are rich source of iron. And any other fruit like apricot, peach, they also have iron in them. And similarly, from meat, we can have iron too, like red meat, meat from goat meat like mutton, beef, or even liver, that is also rich in iron. When it comes to vitamins, so how do these vitamins help us? That vitamins within the body cells, they make coenzymes, and these coenzymes help efficient and faster working of enzymes. So means with the help of these coenzymes, enzyme activity becomes fast and it becomes quick and efficient. So these vitamins are very essential to us. Now we are going to discuss two important vitamins here. One is vitamin D and the other is vitamin C. Vitamin C is very important because it keeps our skin healthy and fresh. It keeps our gums healthy and fresh. And this vitamin C also helps in maintaining healthy immune system. But vitamin C deficiency is there in the food, that we do not have enough food or enough vitamin C. So definitely we are going to suffer from deficiency disease. The major deficiency disease is scurvy. In scurvy, skin becomes rough and cracky, and gums also become loose and they start bleeding. The gums lose their grip over the teeth, and tooth fall is likely to occur. Similarly, prolonged deficiency of vitamin C will lead to poor immunity, and the person is likely to suffer from various infections. But if we want this disease should not affect us, so what we need vitamin C. And what are the sources? Citrus fruit. A fruit that provides us with the vitamin C is orange, lemon. They are the rich source. And even we can have vitamin C from peach and grapes as well. Vitamin D. Vitamin D also helps in maintaining healthy bones, healthy teeth, healthy nervous communication, and healthy muscular contraction. How? Because vitamin D helps promote calcium ion absorption. So if there is enough vitamin D in our blood, in our body, so more and more calcium ion will be absorbed in the body and it will be utilized. But if vitamin D deficiency is there, so definitely calcium ion absorption will be reduced. And again, rickets, we are likely to experience as a deficiency disease, accompanied with poor nervous communication and muscle contraction. So in order to avoid such situation, so we need vitamin D in sufficient quantities in our food, and milk is a rich source of vitamin D, as well as vitamin D can be obtained from the food, obtained from the oceans, obtained from the seas, like fish, prawns, crabs, etc. They can also provide us with vitamin D. And even our skin cells can produce vitamin D when they are exposed to sunlight. So our skin cells are capable to produce vitamin D when they are exposed to sunlight. When it comes to water, so water is very important because living cells and living organisms body, they majorly contain water, making maximum mass of their body. Like within cells at an average have 75% water within their cells. And similarly, living organisms in their whole body, they contain approximately 70% water by mass. Why is that water important? Because water is serving as universal solvent. It's the water that can dissolve inorganic and organic solutes in it. And at a time, more than one solutes can be dissolved within that water. And the best known examples of such solutions, that is blood, and another is milk. Like in milk, we have vitamin D, we have calcium, we have lactose sugar, we have protein casein as well, dissolved within the water. Okay. This water is serving as a cooling agent. Like when we are getting our body hot, when we absorb heat from the surrounding, or we respire a lot. So the water we lose during sweat. When we sweat, we lose water. Water carries excess energies along, and we feel cool when we start sweating. Similarly, in plants, there is a process transpiration. So during transpiration, plants lose water as water vapors that carry excess energies, and plant body is kept cool to normal temperatures in order to stay alive and function successfully. Similarly, water is serving as a reaction medium, and means cytoplasm has got within the cells. Cytoplasm has got a due water concentration. So solids, solutes can dissolve in water, and in their water soluble state, they can collide with the enzymes, they can react, and they can turn into products. Water is needed for photosynthesis as raw material. So if no water, no photosynthesis, no oxygen, no food available, not even for plants, but for animals as well. At the same time, what we have water important to us because hydrolysis, that is digestion that occurs inside our digestive system. So this digestion is also mainly hydrolysis that needs water because enzymes that are responsible for digestion of protein, they have got two active sites. One active site contains protein, and the other active site can be loaded with water. So without water, food cannot be digested. Water is serving as transport agent. Water that is rising up to the leaves from the roots, that also carries minerals in the dissolved form. Even the sugars prepared, amino acid prepares in the leaves during photosynthesis. So these sugars are traveling in the whole plant body in the water soluble state. The blood that is running in our blood vessels that carries these nutrients in the water soluble state from one part of the body to another part. And water is important for excretion because the urine we excrete, that mainly contains water because the toxic substances we are getting rid of, they are highly toxic, and this water dilutes them, and this water actually basically helps us to get rid of these waste substances. Water is very important for the aquatic organisms like fish, like tortoises, like prawns, all other underwater animals, even underwater plants, they need water as their immediate environment. So water is important for them because they maintain all their needs with the help of water that is surrounding them. So if there is not enough water in our body, what problems we can face? Dehydration, and dehydration will make us sluggish, will make us do little work, and will not let us work or live effectively. At the same time, less water means we may be suffering from constipation problem, and prolonged constipation can lead to intestinal ulcers. At the same time, toxicity would be prevailed within the body because less water, less urination, so toxic substances like urea would be staying more of the time within the body. So problems would be raising for the body as well. And where we can have water, means we can directly have water by drinking water or water made juices or shakes. Food can also provide us with water, and even when we respire, so that also produces water and that gets mixed up within the body. Dietary fiber. Dietary fiber, generally known as roughage. This dietary fiber is very important to us because it keeps our intestine healthy, it prevents constipation, and it reduces the chances of developing intestinal cancer. What is this dietary fiber actually made up of? Plant products, they do contain cells, and plant cells do have cell wall made up of cellulose fibers. And these cellulose fibers are actually serving as roughage, serving as dietary fiber. We do not produce any enzyme that can digest cellulose fibers. So cellulose fibers remain in our food residue, or in our feces as undigested material. But this is a benefit to us. Why? Because cellulose fibers have got strong affinity for water molecules, and it retains some water inside the feces, inside the residue. It does not allow the intestine to absorb all the water in the residue. Some of the water is retained within the residue, and this water keeps the residue, keep the feces soft, moist, and for intestine, it becomes easy to push that food in the forward direction towards rectum, and then later pushing it out of the body. But if there is no water, so that will lead to constipation, because cellulose fibers are not there in the diet, they are not there in the residue. Intestine will absorb all the water in the residue will become dry, and it will become hard. And it would be harder for the intestinal muscles to push that residue, that feces, forward towards the rectum. And in that case, the friction that is exercised by that dry residue, that friction can cause ulcers on the wall of small intestine, even on the wall of large intestine. And these ulcers can, in future, can lead to intestinal cancer. That is what we generally know as colon cancer. So deficiency of dietary fiber will lead less water in the residue, that will lead to constipation, intestinal ulcer, or colon cancer. So that's why what we need to feed on plant products like fruit, like vegetables, etc. So these are the few components of the diet that are taken as micronutrients, like minerals and vitamins we discussed. And we have also discussed about water and dietary fiber significance. And next we are going to discuss about carbohydrates, proteins, and fats and their significance. Among macronutrients, one essential nutrient is carbohydrate. Carbohydrates are organic molecules made up of three basic elements: carbon, hydrogen, and oxygen. And they are very, very essential to us. Carbohydrates like glucose is the simplest carbohydrate, and it is needed for respiration. 5-carbon sugar like ribose and deoxyribose are needed for making nucleic acids of DNA and RNA. Cellulose is also carbohydrate that is required for making cell wall in plant cells. And similarly, starch and glycogen are the large carbohydrates that are stored within the cells as higher reserves for glucose. When we talk about carbohydrates, so carbohydrates are of three types. One is known as monosaccharide. Mono means one, saccharide means sugar. Carbohydrates that are consisting of single sugar unit, these are known as monosaccharides. And the monosaccharides include glucose, fructose, galactose, and all these three molecules have got the same chemical formula, that is C6, H12, and O6. And these three molecules are isomers to each other. Other monosaccharides include ribose, needed to make RNA, deoxyribose, needed to make DNA. The main source of energy to us is glucose, that is respired, whether aerobically or anaerobically, and this is glucose. If we eat on plant products, we obtain starch. If we eat on animal products, we obtain glycogen. And when this starch or glycogen is hydrolyzed within our digestive system, So they release glucose that is transported to each and every body cell, where cells take them and respire on this glucose. If we want to confirm whether our food does contain glucose or not, because the food that is sweet in taste is likely to contain glucose. So if we want to confirm that our food does contain glucose and not, so we can conduct glucose tests, and this is also known as Benedict test, or reducing sugar test, or simplest sugar test. So what we do, we take a test tube. In that test tube, we add 5 cm cube of sample solution that we are expecting to contain glucose, and we add 5 cm cube of Benedict reagent. Benedict reagent has got blue color, so when it is added to the test tube, so the whole mixture turns blue. So we heat that mixture in the test tube in the water bath, approximately at 90 degrees Celsius temperature for approximately 180 seconds, and we observe the color change. If after heating in the water bath at 180 degrees Celsius, it does not change color and the color of the test tube contents remain blue, it means no glucose, no reducing sugar. But if the color changes to green, it means very minute concentration of glucose available. If concentration changes to yellow, so it means it has got small concentration of glucose. And if the test tube contents gain orange color, it means high concentration of glucose. Or if they gain brick red color, it means very high concentration of glucose. So this Benedict test is not only the qualitative test, it is quantitative test as well. It ensures us that sample contains glucose if the color appears to be green, yellow, orange, or orange red. And these different colors also give us an idea of the different concentrations of glucose in the given food sample we have tested with the Benedict reagent. Then comes disaccharides, mean carbohydrates that are consisting of 2 sugar units, means 2 monosaccharide units chemically bind together to make a disaccharide molecule. A disaccharide molecule is the larger molecule. Like 2 glucose units will chemically bind to make 1 maltose molecule. And similarly, glucose and galactose will bind to make lactose molecule. And glucose and fructose can bind to make sucrose molecule. So sucrose, lactose, and maltose, these are the 3 disaccharides. And these have got the same chemical formula, that is C12, H22 and O11. And these 3 can be taken as isomers to each other. And when these disaccharides are hydrolyzed, they will be yielding the same monomers that they are made up of. Same monosaccharides they are made up of. Like when lactose that is found in the milk, it is hydrolyzed, it is broken down, it releases glucose and galactose. When sucrose is hydrolyzed, it releases glucose and fructose. And when maltose is hydrolyzed, it releases 2 molecules of glucose. Remember one thing, like monosaccharides, as monosaccharides are sweet in taste, water soluble and diffusible, so these disaccharides are also sweet in taste and they are also water soluble and diffusible. Now, among the disaccharides, maltose and lactose are taken as reducing sugars because when they are heated with Benedict reagent, they directly give positive result. But sucrose is not taken as the reducing sugar, rather it is taken as non-reducing sugar because when it is directly heated with Benedict reagent, it does not give positive result. So in order to confirm the presence of sucrose, we need to carry on a test that is known as non-reducing sugar test or sucrose test. This sucrose test, what we do, we take a test tube, we add 5 cm cube of the given sample solution, that is the sucrose sample, and we add 5 cm cube of dilute hydrochloric acid solution. We heat the mixture in the water bath near to boiling. Then we take out the test tube, cool it down to the normal temperature around, and then we add alkali 5 cm cube in that mixture. After that, we add Benedict reagent and we heat it in the water bath at 90 degrees Celsius temperature constantly for around 300 seconds and then observe the color change. Now, how these different treatments will affect, like heating the sucrose solution with the dilute hydrochloric acid, it will break down sucrose into its monosaccharide units like glucose and fructose. Treating the mixture with alkali will neutralize the acid, so that acid must not react with the Benedict reagent. And now the Benedict reagent will give us the positive result. If we have yellow color of the test tube contents, that means small concentration of sucrose. And if the test tube contents turn orange red or brick red, it means high concentration of sucrose. The third type of carbohydrates is polysaccharides. Poly means many, saccharide means sugars. So carbohydrates that are made up of many sugar units, these are known as polysaccharides. And polysaccharides are mostly flat in taste, means tasteless. They are insoluble, they do not dissolve in water, and they are indiffusible, means they cannot diffuse as well. And we have got the three well-known polysaccharides, that is starch, glycogen, and cellulose. Remember one thing, when these polysaccharides are hydrolyzed, they are digested or they are broken down, they release the monosaccharide units they are made up of, like starch, glycogen, and cellulose. These three polysaccharides are made up of glucose. So when they are hydrolyzed, they are likely to yield glucose. Starch is the storage molecule, higher reserve for glucose stored in the plant cells. Glycogen is the higher reserve of glucose stored in the animal cells. And cellulose molecule makes cellulose fibers that make up cell wall of the plant cells. And all these three carbohydrates, polysaccharides, are made up of glucose. Our food contains starch, that is a major source to us, and that makes the maximum bulk of our food. So this starch can be found in potato, wheat grains, rice grains, etc. So if you want to confirm the presence of starch, so we conduct iodine solution test, or this is also known as starch test. So what we do, we take a sample solution in a test tube and we add 1 or 2 drops of iodine solution. Iodine solution has got yellow brown or golden brown color. So when this is mixed with the starch solution, so iodine reacts with starch and it turns the contents blue-black. So blue-black color of the test tube contents is the confirmation that the sample contains starch. So this was the discussion about carbohydrates. We discussed about monosaccharides, disaccharides, and polysaccharides, and we discussed about their lab test as well that will help us to identify their presence and their concentration in the given samples. Now we proceed to lipids. Lipids are the organic molecules and they are also made up of 3 elements: carbon, hydrogen, and oxygen. Lipids have got very important functions within the cells, like membranes, including cell membranes. They are actually made up of 2 layers of lipids. Similarly, lipids present as adipose tissue, so they insulate the body. Lipids are required for making certain hormones, like there are 3 important hormones in the human body, like in males testosterone, in females estrogen and progesterone. These are the hormones that are made up of made up of special lipid molecule known as cholesterol. For certain vitamins, lipids are required, like vitamin A, vitamin D, and vitamin E. They are functioning well when they are dissolved in fats, when they are dissolved in oils. These oils and fats are 2 different types of lipids. Lipids are chief source of energy. Oils and fats can be respired in the body and they can provide us with more energy when we compare with the proteins or the carbohydrates. Cuticle that is found on the surface of epidermis in case of leaves in plants. So this is also made up of wax, and this wax is actually a lipid. A lipid molecule is made up of three components. One is glycerol, that is a special type of alcohol, and it binds with three fatty acids. And then we have a lipid molecule, and this lipid molecule is also known as a triglyceride. A lipid molecule represents all the properties of the lipid it is a part of. So different lipids, like different oils or different fats, they have got different molecular structure that is different from each other. So when a lipid molecule is hydrolyzed, it will be broken down and releases three fatty acids and one glycerol. If we want to confirm the presence of lipids in a diet in a given sample, so we will have to conduct the iodine solution test. But before going to the iodine solution test, there is another segregation of lipid that I would like to discuss. That is, lipids are generally classified into two types. One is known as saturated lipids. These are generally known as fats, and at room temperature they are found in the solid or semi-solid state, and they do contain maximum energies. And the examples are margarine, animal fats found in the animal body, or cream found over the surface of the milk, butter, or even ghee. So these are the examples of saturated lipids. They are generally known as fats. The second type of lipid is oils that are liquid at room temperature. They comparatively contain less energies, and these oils are mainly from the vegetable sources, like vegetable oils like canola oil is there, sunflower oil is there, corn oil is there, or even olive oil is there. So these are all the examples of unsaturated lipids that are known as oils. Oils can be converted into fats by the process of hydrogenation on the industrial scale. In order to confirm the presence of lipids, that is either in the form of fats or either in the form of oil, so we can conduct a test known as ethanol emulsion test. So what we do in that test, if we have the liquid sample, we take 5 cm cube of it, we add 5 cm cube of pure ethanol into that sample. We shake it well, and then we add 5 cm cube of the distilled water and shake well. What we observe, we see the test tube contents have turned cloudy and a foamy emulsion may appear over the surface. So this is the confirmation of lipids in the given liquid sample. But if we have a solid sample like peanuts, like almonds, like cashew nuts, or any other source, so what we do crush it first, grind it to powder, add that powder in the test tube, add 5 cm cube of pure ethanol and shake it till the whole powder is dissolved in the ethanol. Then we add 5 cm cube of the distilled water in the same test tube, shake it well, and we find the same results like cloudy test tube contents and foamy emulsion on the surface. That confirms the presence of lipids in the given sample. Proteins, another type of macromolecules, macronutrients found in our diet. And these proteins are very important to us. These proteins are made up of 4 essential elements like carbon, hydrogen, oxygen, and nitrogen. And apart from these 4 essential elements, some other elements can also be found, but depending on different types of proteins. Proteins are very important because most of the enzymes, means all the enzymes that catalyze chemical reactions inside our body, within the cells, within the organelles, so they are all made up of proteins. Most of the hormones like insulin, like ADH, they are made up of proteins. At the same time, pigments like hemoglobin found in the red blood cells, that is responsible for the transport of oxygen, and similarly chlorophyll, that is found in the mesophyll cells, in the chloroplast of the mesophyll cells in the leaves, that is also a pigment. So they are protein pigments. Without chlorophyll there is no photosynthesis. Without hemoglobin there is no oxygen transport. Similarly, these proteins means muscle proteins like actin filaments and myosin filaments responsible for muscle contraction. These are proteins. And we have proteins as storage molecules as well, like casein that is found in milk is the protein store, and albumin found, that is also known as egg white, in the eggs, that is also a protein store. So proteins have got vital importance, and if we do not have any carbohydrate or fat reserves in our body cells, in our body tissues, so we can still respire on proteins, respire on amino acids. Just like glucose is the simplest unit of all the carbohydrates, so similarly, amino acid is the simplest unit of all the proteins. There are 20 different types of amino acids, and these amino acids can bind together in different number, in different sequence, in different ratio to give rise to different types of proteins. In our body, we can have a protein made up of hardly 9 amino acids, and we can also find a protein that is made up of more than 3,000 amino acids. But the type of amino acid used to raise different types of proteins is the same, that is the 20 different amino acids are there. These amino acids chemically bind together to make polypeptide chains, and these polypeptide chains can raise the larger protein molecules that serve various functions in the body. If we want to confirm the presence of protein in the given food sample, so what we conduct a test that is known as biuret reagent test or protein test. In this biuret reagent test, what we do, we take a test tube, we add 5 cm cube of the sample solution in that test tube, and then we add 5 cm cube of the biuret reagent. It is also known as biuret solution. This biuret reagent has got blue color, and all the test tube contents will turn blue. We will shake the test tube contents and we will rest it for a minute. And after a minute, we see the color change in the test tube contents. And if the test tube contents turn purple, that could be taken as purple, mauve, violet, or even indigo. Light purple color will reflect low concentration of proteins, and dark purple will reflect high concentration of proteins. But if color does not change, it remains blue, it means no proteins at all. Balanced diet. A diet that contains all the nutrients, and these nutrients are present in the ratios, in the proportions required by a living organism, required by a human. A balanced diet contains all the nutrients, and the nutrients are present in a specific ratio. And this specific ratio is desired or needed by that human, by that person. And this ratio, or you can say the ratio of these nutrients in the balanced diet, can vary from person to person. And this variation depends on lifestyle, age, and gender. Like a 25 years old male laborer needs more energy, more carbohydrates, more fats, because it has to do a lot of hard work moving things from one area to another area, compared to the same age, same gender office worker. So you can even say a 25 years old male marathon runner would be needing more energy than 25 years old male teacher who has got less activity level. So lifestyle can vary the demand of energy. Accordingly, energy yielding components must be present in the balanced diet. Similarly, when we talk about females, a 25 years old female needs more iron compared to 60 years old female. Why? Because 25 years old female is experiencing menstrual cycle, and she is losing blood in every menstrual cycle. In order to compensate for the loss of that blood, so she needs more iron in her diet compared to the 60 years old lady who is experiencing menopause and is not experiencing menstruation at all. When it comes to age, again, it makes a difference. Okay, means just the example I told you right now, so that is also reflecting the age as well, because 25 years old female is younger, is more active, and needs more energy and more iron compared to 60 years female. And even you can see that 25 years old female could be office worker, could be an athlete, and the 60 years old female would be most of the time spending her time at home. And even if we go with the gender, like 25 years old female would be needing less energy compared to 25 years old male, because males are more mobile, more active, so they need more energy compared to females. You can yourself experience that the playgrounds like football ground, hockey ground, or even cricket ground that are set for female players, they are even smaller compared to the ones that are set for the male players. So that is actually on their energy level. So the balanced diet, one thing I repeat, balanced diet has got all the nutrients, and the nutrients are in the desired proportions, and the proportions desired is based on the lifestyle of the individual, on the age of the individual, and based on the gender of the individual. But not everyone is able to maintain the balanced diet in his daily dietary routine. If we do not take a diet with the balanced proportion, so this diet is taken as malnutrition. So malnutrition refers to intake of a diet that does not contain the nutrients in the balanced proportion. So this is known as malnutrition. And malnutrition can be categorized into like undernutrition and overnutrition. Undernutrition means one or more of the nutrients in the diet, they are present in less quantities than they are required. And the persons using such diets, they will be suffering from deficiency disorders. In case if a person is consuming a food that contains less iron, so that person is likely to suffer from anemia. If a person is using a food that contains less vitamin C, so that person is likely to experience scurvy. If a person is taking less protein, that person is likely to suffer from kwashiorkor or marasmus. And then it comes to overnutrition. If a person is consuming a food that contains one or more nutrients in larger quantities than what he actually requires, so that excess intake of certain nutrients can also create problem, can also cause abnormalities too. For example, excess intake of fats and sugars in the daily diet will result obesity, will result heart attack, chances of heart attack will increase, chances of stroke will increase, and blood vessels are likely to get blocked as well because of excess fats traveling through those blood vessels. And these people are likely to suffer with high blood pressure. So excess intake of any nutrient in the diet that is taken as overnutrition, that is also one of the category of malnutrition, and that is problematic to life as well. So we all need to have a balanced diet, and we all need to check what are our energy requirements of the day, and accordingly we must set our dietary chart. But in certain parts of the world, people do not find proper food. Forget about the balanced diet. They find it very hard to meet their daily energy requirements, their daily diet. This is what we call famine. Means people are suffering from famine in certain parts of the world, like in certain countries, in Africa, certain parts of the Asia, and the areas of the world, they are constantly experiencing wars like Syria, like Palestine. So in these regions, the people are facing a lot of difficulty to meet their dietary requirements. So these people are suffering from famine. What are the causes of famine? Consistent wars in certain areas will not make these people to grow any crops, to carry any businesses, to earn any salaries or wages. or any revenue from their businesses. So they could be suffering from famine. Countries constantly in wars, so they are unable to maintain their financial reserves to feed their people. Similarly, natural disasters like floods, hurricanes, earthquake, so they also destroy the vast areas. They also destroy the crops and the agriculture lands, and again there is little food. Similarly, locust attack on the fields can also deprive us with the food. So natural disasters, wars, poverty, poor economic state of the country will lead to famine. And the areas that are striking famine, that are experiencing famine, have to rely on the food aid provided to them from certain countries of the world, certain capitalists of the world. And they do not provide them with the balanced diet. They provide them with the diet that can meet their basic requirements. Like in the famine-struck areas, people might be having food, but food is not balanced. And likewise, they are suffering from deficiency disorders, because if their diet is deficient in iron, they would be suffering anemia. Is deficient in protein, they would be suffering from kwashiorkor or marasmus. So we need to secure health in these regions by providing them with the balanced diet. We are going to discuss about human digestive system. In this segment, we will discuss about certain terminologies being used in the digestive system. We will discuss about the teeth, structure of the tooth, and how the teeth are involved in the mechanical digestion, as well as we will be discussing about chemical digestion that occurs in mouth and stomach. So human have got a holozoic mode of nutrition, means they do have a tube-like digestive system with two openings. One opening, that is mouth, and through this opening food enters the digestive system. Then it is processed gradually from different components of this tube, and at the end of the tube there is a second opening, is known as anus, and through this anus undigested food residues are expelled in the form of feces. Certain terminologies that are likely to be discussed today, that include ingestion. Ingestion means intake of food into the digestive system, that mainly occurs at mouth. Then comes digestion. Digestion involves the breakdown of food, and this breakdown of food is actually involving two types of digestion. One is known as physical digestion, or what we call mechanical digestion. In case of physical or mechanical digestion, larger food pieces are broken down into smaller food pieces. And remember one thing, no enzyme or no chemical activity is involved here. And it is a simple process where larger food pieces are broken down into smaller food pieces, and molecular status of the food remains unaffected. But it helps in increasing the surface area of the food, and it also helps the enzymes to work more efficiently and fastly to digest the food. Then comes chemical digestion. In case of chemical digestion, enzymes are involved, and larger, complex, and mostly insoluble food molecules are broken down into smaller, soluble, and simpler molecules that could be taken up by the cells and utilized as per their need. So in that case, enzymes are involved, and complex molecular status of the food is converted into simplest molecular status of the food. So this process is actually occurring in mouth, in stomach, and in small intestine of the human digestive system. Then comes absorption and assimilation. Absorption means digested substances are taken up from the small intestine into the bloodstream, and they are circulated throughout the body, where body cells take up these components, these digested molecules, and utilize them as per their need. Like glucose is used for respiration, amino acids are used for making different types of required proteins. And assimilation is actually utilization of the food or digested food into the cells. Then comes egestion, and egestion is the removal of undigested food residues through anus. that is the second opening, and this egestion actually helps getting rid of the undigested residue so that digestive system is ready for another food intake. So let's continue with our discussion regarding different components of the digestive system. Digestive system comprises of the following components: mouth, esophagus, stomach, small intestine, large intestine, rectum, and anus. Some of the authors have included rectum and anus as the component of the large intestine. So it is not a big deal at all. These are the connected organs of the digestive system. They are connected in a series, and all these organs take up food and take up food residues turn by turn and process it as they are specialized for. But there are two other organs, and these organs are liver and pancreas. These are the organs that are connected with the small intestine. These organs do not directly take up food and process it. Rather, they produce and release secretions into small intestine, and these secretions are either helping the food digestion process to complete and effectively take place, or these secretions are actually containing enzymes that are directly involved in the process of digestion. So we can call them as supporting digestive organs, or organs that are supporting the process of digestion. Let's start with the mouth. What happens in the mouth? What processes are occurring in the mouth? Mouth is the first organ of the digestive system, and the two main processes that occur in the mouth are ingestion and digestion. Ingestion means the food is taken into the digestive system through mouth, and digestion means food is broken down and partially processed within the mouth. So there is two types of digestion that is occurring in the mouth. One is known as the chemical digestion and the other is mechanical digestion. First of all, we will discuss about mechanical digestion, also known as physical digestion, where teeth with the help of tongue and cheeks break down larger food pieces into smaller granule-like chunks, and this is increasing the surface area of the food so that enzymes are going to work more efficiently on the partially broken down food particles. So larger food pieces, larger food particles are broken down into smaller food pieces so that enzyme activity becomes very effective. So chemical digestion involves saliva. Saliva is a liquid that is produced by the salivary glands in the mouth, and this saliva contains mucus that liquefies the food, and this saliva also contains an enzyme. This enzyme is known as salivary amylase. This salivary amylase digests starch that is present in the food, and the starch is digested into simple sugars that is also known as maltose. Remember one thing, in mouth only starch digestion does take place. No protein and no fat digestion is taking place in the mouth. Why? Because in the mouth only salivary amylase is produced. No enzyme responsible for fat or protein digestion is produced. So means there is no enzyme that could digest fats or proteins in the mouth. So only starch digestion does take place. Remember one thing, the pH of the saliva is 7, that is the neutral pH, and salivary amylase is capable of working effectively in the pH range, that is 6.5 to 7.5. But if pH increases above 7.5 or if pH decreases below 6.5, so salivary amylase gets denatured and it no longer digests starch. Within mouth, by the activity of teeth, larger food pieces are converted into smaller food pieces, and then later salivary amylase digests starch into simple sugars. But remember, not all the starch is digested here. Only a little portion of starch is digested here because food stays in the mouth for a little time. Now, that partially processed food is converted into pellet-like structures.
and these pellet-like structures are known as bolus. Singular, it is known as bolus, and this bolus is swallowed into the esophagus. Before we proceed further with the esophagus, what happens with the food when it travels through esophagus towards stomach, we must discuss about teeth. Teeth, the structures, the bony structures that are responsible for mechanical digestion in the mouth, we must know about these teeth. There are 4 different types of teeth found in the human mouth, and these teeth include incisors that are meant for cutting and biting, canines that are meant for cutting and tearing, premolars and molars that are responsible for grinding, for chewing food into smaller granule-like structures, granule-like pieces. So these 4 different types of teeth are arranged in 2 jaws, one the upper jaw and the other lower jaw, and there is a sequence of these teeth in the human mouth. Remember one thing, we are discussing about a human mouth, and we are discussing about the teeth found in the human mouth. But considering the different other animals, herbivores means the animals that are feeding more on plants or plant products. So these animals have got very much prominent incisors in the large number, and canines are either absent, or if present, they are smaller in size. But premolars and molars would be large in number. Whereas the animals who are likely to feed on flesh, feed on the muscles of other animals like carnivores, like cats, dogs, alligators, etc., lion as well. So they have got very less number of incisors, very less number of premolars. A few molars are found, but canines are very much prominent because they have to tear the flesh of the animal, so canines are very much prominent. So number of the teeth and the type of the teeth and their development within the mouth depends on the eating habits, genuine eating habits of the animals that teeth are found in. So these teeth are well inserted in the jaws. The jaw bones, the U-shaped jaw bones, these teeth are well inserted in the jaw bones, in the tooth sockets, and they are well protected and covered around by the gums. Now we will consider the structure of a tooth. A tooth is consisting of 2 parts. One is known as the root. With the help of this root, this tooth is well inserted in the tooth jaw and is well covered, well protected, and well supported by the gums around. And the crown, that is the exposed part of the tooth, and that what we see, that the white part of the tooth that we see, that is known as the crown. Now the crown contains the topmost layer, that is known as the enamel. Middle bony part is known as dentine, that gives it hardness. And the inner is known as pulp, that is the fleshy part, that contains the nerve endings, and that also contains certain sensitive receptors. And these are the nerve endings. This is the pulp when it is exposed to the different chemicals in the food, so that imparts sensitivity. And these nerves are entering the tooth through the roots, and then they are becoming the part of the nervous system. So this is the general structure of a tooth. In humans, generally in the life cycle of the human, or in the whole life of the human, 2 sets of teeth are found. In the early age, by the age of 6, the set of teeth found in the human mouth, that is known as the milk teeth, that are the temporary teeth, and that fall off and they are replaced at the age of 6 or above with the permanent teeth. And permanent teeth last throughout the life, or due to some other reasons like decay or some other reasons, they could be falling off, but they are less likely to be replaced by the new teeth. Now, there is a concept of tooth decay. This is not exactly required in general for the Cambridge students, but actually it must be the common understanding and common learning of all the students, that they should know about tooth decay and they should know how to prevent tooth decay. So it's the general information that should be considered by each and every student. When we eat some food that is rich in sugars, that is rich in starch, rich in carbohydrates, so bacteria in our mouth that are likely to metabolize the pieces of the food or the molecules of the food that remain trapped within the gums, between the teeth, or somewhere in the mouth. So these trapped particles are metabolized by the bacteria living in our mouth, and they produce organic acids. These organic acids, first of all, they destroy the enamel, then they destroy the dentine, and the pulp is exposed. And when the food particles, they approach the pulp, so sensitivity in the form of pain does reflect. And then while eating something, drinking something, we may feel pain because the pulp is exposed and food molecules are interacting the pulp. In order to avoid such condition, what we need to do? We need to brush our teeth regularly with the fluoride toothpaste, because fluoride strengthens the enamel. We must avoid eating sugar-rich food, or if we do eat, we must rinse our mouth properly with plenty of water. We must use antiseptic mouthwash that will reduce the population of bacteria in our mouth. And we must visit the dentist once or twice per annum. So if there is any drawback or any damage that has started occurring to the teeth, so the dentist will observe it and would counter it immediately before the damage occurs very severe. So that was about the structure of the tooth and how the teeth are involved in mechanical digestion. Now we come across with esophagus. The food bolus that was made within the mouth is swallowed into the esophagus. Esophagus is the muscular tube, and this muscular tube is lying at the floor of the mouth cavity, that is also known as the buccal cavity. And this esophagus is actually responsible for transporting food, transporting bolus from mouth to the stomach. So in that case, the muscles, they produce a wave of contraction in the esophagus wall that is known as peristalsis. And how does it happen? That when the food enters the esophagus, the muscular wall that contains two types of muscles, longitudinal muscles and the circular muscles. So these muscles constrict at the back of the bolus and dilate in front of the bolus. And they produce a wave of constriction that starts its journey at the start of the esophagus and continues, this constriction wave continues till the bolus reaches to the stomach. So this constriction wave that pushes the bolus in the forward direction from the mouth towards the stomach, this is known as peristalsis, or it is known as peristaltic movement. Remember one thing, esophagus does not produce any enzyme that could digest food components. Means food contains starch, proteins and fats. So there is no enzyme produced in the esophagus that could digest these food components. But a little starch digestion continues to occur in the esophagus. Why? Because the bolus that was formed in the mouth, it also contains saliva that is mixed in the bolus. And within that saliva, there is salivary amylase. So that salivary amylase keeps on digesting starch throughout the route when the food is traveling towards the stomach. At the junction where esophagus joins the stomach, there is a ring of muscles, or you can say there is a valve, and this valve is made up of ring of muscles, and this is known as cardiac sphincter. This valve allows the food to enter from esophagus to the stomach, and this ring opens when the peristaltic wave reaches to the cardiac sphincter, and it allows the bolus to fall into the stomach, and then later it again closes. Now this cardiac sphincter, it is named because this particular valve area is close to the heart, so that's why this is known as the cardiac sphincter. Now the food bolus has reached the stomach cavity. Stomach is a muscular organ, and this stomach is also a tube-like organ where the partial food digestion does take place, but here protein digestion is likely to occur. In the stomach, it consists of various tissues. For example, stomach contains epithelial tissue. Outer epithelial tissue are known as outer epithelium, that makes the outer boundary, and the inner epithelium makes the inner boundary that is facing the cavity of the stomach. Then this stomach contains muscles, and these muscles are likely to produce peristaltic movements that will push the food forward into the small intestine when it is required. And these muscles can also produce blending movements, or what we call churning movements. And these churning movements are actually responsible for blending the food contents appropriately, precisely with the gastric juice in the stomach. Then we have got glands within the stomach wall. Glands are specialized cells found in the wall of the stomach. And these glands are likely to produce a secretion, and this secretion is known as gastric juice. This gastric juice contains a number of components. For example, the first primary component of the gastric juice is mucus. Mucus actually it liquefies the food further when it gets mixed up with the bolus, and at the same time this mucus also makes a protective coating over the inner epithelium in order to prevent it from the acids, from the effect of acids, and from the effect of protein digesting enzymes. Then we have HCl, hydrochloric acid. Hydrochloric acid is produced by the gastric glands with a strong pH of 2, and this hydrochloric acid performs a number of jobs. Number one, it kills all the germs that enter the digestive system along with food or drink. Second, this hydrochloric acid helps maintain acidic pH within the stomach that will help in activating enzymes, and that will also help in the enzyme activity efficiently. This hydrochloric acid also tenderizes the food proteins, means larger food polypeptides, or you can say larger food proteins. They are broken down into smaller polypeptides so that enzyme activity becomes faster and efficient. And the fourth job of this hydrochloric acid is that it is also responsible for activating the enzymes. I repeat, in stomach only protein digestion occurs. No carbohydrate, no starch, no fat digestion, because gastric glands only produce protein digesting enzymes. They can also be taken as proteases. They can also be known as proteases. So these proteases are of two types. One is known as pepsin, that is the active form, and inactive form is pepsinogen. This pepsinogen, once it is produced and it enters the stomach cavity where it interacts with hydrochloric acid, it gets activated and it transforms from pepsinogen into pepsin. And this pepsin is likely to break down larger protein molecules into smaller polypeptides or smaller peptones. Whereas another enzyme that is known as prorenin, that this prorenin is an inactive form of the enzyme. When it enters the stomach cavity, it interacts with the hydrochloric acid, it becomes activated as renin, and it is responsible for digesting milk proteins and for curdling of the milk. So only these two enzymes that are processing proteins from larger molecular status to smaller polypeptide status, this is occurring only, and this is not the complete protein digestion. It is incomplete protein digestion. Whereas no enzyme is produced that could digest starch or fats. And even the salivary amylase that was already present in the bolus along with saliva, so that salivary amylase also gets denatured when it reaches the stomach cavity and it encounters with the hydrochloric acid. So it is denatured. So starch digestion that had started in the mouth, that continued in the esophagus, would stop in the stomach. Now in the stomach, mechanical digestion is partially done by the activity of muscles because they do produce churning movements, and this churning movement can be taken as a partial mechanical or physical digestion. And this is further supported by enzymes in the gastric glands that actually carry on chemical digestion, but only on the proteins. So the partially processed food in the stomach is now known as chyme, and this chyme is almost liquid. Even though you have taken up the solid food, now that solid food has been turned into a semi-liquid chyme, and with each peristaltic wave, this semi-liquid chyme with a little portion of the semi-liquid chyme is pushed into small intestine with each peristaltic wave. I repeat, where each peristaltic wave results, a little portion of chyme is introduced into the small intestine. At the junction of stomach and small intestine, there is present another valve, and this valve is also made up of ring of muscles, and this is known as pyloric sphincter. So the peristaltic wave that started its journey at the start of esophagus, this ended up at the cardiac sphincter, but the peristaltic wave that started its journey from the start of the stomach, when it reaches to the small intestine end, it makes the pyloric sphincter to open, and a little chyme is pushed into the small intestine where it is further processed. We are going to discuss about the small intestine, its role in digestion, absorption, and then we will be discussing about the role of liver, pancreas in support of digestion process, and role of large intestine. So let's continue with small intestine. Small intestine is the muscular tube that has approximate length 6 meters, and this muscular tube receives acidic chyme from the stomach. At pyloric sphincter, stomach continues to small intestine, and at each peristaltic contraction wave, a little portion of acidic chyme is introduced in small intestine. Small intestine is a lengthy tube of about 6 meters length, but it is divided into 3 segments: duodenum, jejunum, and ileum. And all the 3 segments are playing their significant role in the process of digestion and absorption. Small intestine is very important in the digestive system because it is the only organ in the digestive system where all the food grades are not only digested, but also the digested substances are absorbed into the bloodstream and transported across the body. Duodenum receives acidic chyme. This acidic chyme contains HCl, and this is possible that this HCl is likely to denature all the enzymes that are to digest food in the small intestine. Why? Because in small intestine, enzymes need alkaline environment, but the chyme contains HCl that is acidic, so it would likely denature the enzymes. So no digestion is likely to take place in small intestine. So this is naturally prevented by the secretions coming from liver. Liver produces bile that is stored in the gallbladder, and when the digestive activity starts in the duodenum right after receiving chyme, so a little bile is introduced through bile duct into the duodenum part of the small intestine. This bile contains minerals, pigments, but does not contain any enzyme. It does not take part in digestion of food directly, but it creates the environment that makes the food digestion completed in the shortest time. The minerals present in the bile, they neutralize the acid present in the chyme. These minerals also help in defining the alkaline pH in the small intestine cavity. This bile also activates enzymes that are coming from pancreas, and this bile also emulsifies fats. The fats, they are emulsified from larger fat droplets, from larger fat globules to smaller fat droplets or globules. This increases their surface area, and it helps the lipase enzyme to digest lipids, digest fats in the shortest time. Now the duodenum also receives secretions from the pancreas through the pancreatic duct. Pancreas produces three important enzymes. One is lipase, second is amylase, and the third is trypsin. All the three enzymes are produced in an inactive form and they are released as pancreatic juice into small intestine through pancreatic duct. This pancreatic juice has got alkaline pH because all the three enzymes are to work in the alkaline environment, and the same alkaline pH around 8 and 9. In some of the books it's written 8.5. In some of the books authors have demonstrated it as pH 9. But both are equally effective pH. So these enzymes have to work in the small intestine. These enzymes are produced in an inactive form like trypsin. It is produced in an inactive form known as trypsinogen. When this trypsinogen reaches the small intestine and interacts with the bile, all already pre-activated enzymes, it gets activated to trypsin and it is likely to digest proteins into dipeptides and amino acids. Similarly, prolipase is the inactive form of enzyme. When it gets encountered with the bile in the small intestine, it gets activated into lipase and it digests emulsified fats into fatty acids and glycerol. At the same time, amylase that is produced as proamylase, it gets activated when it interacts with the bile in small intestine, and this amylase digests remaining starch into maltose. But remember one thing, these are not the enzymes only added up by the pancreas, but intestine does contain some glands. These are known as intestinal glands and they produce a secretion known as intestinal juice. This intestinal juice does contain mucus that makes a protective coating around the inner wall of the small intestine. At the same time, this mucus further adds liquidity to the food that is being processed in the small intestine. Along with the mucus, these intestinal glands produce some enzymes, like they produce an enzyme known as maltase that digests maltose, a disaccharide, into glucose. Another enzyme, sucrase, that breaks down sucrose molecules into fructose and glucose. And another enzyme, lactase, that breaks down lactose present in the milk into glucose and galactose. Remember one thing, lactose, maltose and sucrose, these are disaccharides and these are not absorbed into the cells. So they are to be broken down into their simpler monosaccharide units, and that is done by the intestinal enzymes that I have named earlier. Similarly, another enzyme that is produced by intestinal gland, that is known as dipeptidase enzyme, and this dipeptidase enzyme breaks down dipeptide molecules into amino acids. Dipeptide molecules are shorter proteins consisting of only 2 amino acids. So this dipeptidase enzyme breaks down this dipeptide molecule into amino acids. Now in the small intestine we have got absorbable products, and these products include glucose, fructose, galactose. Fructose and galactose are isomers of glucose, so that's why we will mainly discuss glucose. So glucose, fatty acids, glycerol and amino acids are the digested products of carbohydrates, fats and proteins that are to be absorbed into the bloodstream and circulated into the body. But along with these substances, certain minerals, water and some vitamins are to be absorbed in the small intestine as well. Small intestine has got 3 segments, as I told you earlier. Duodenum is the first few inches of the small intestine where digestion process starts, and that reaches at its peak in the jejunum part where digestion and absorption reaches to the peak. In the last part, absorption of the digested substances along with some minerals, some water and some vitamins, particularly vitamin B12, they are absorbed into the bloodstream. In order to absorb these substances efficiently, inner wall of the small intestine is projected into finger-like outgrowths. These finger-like outgrowths are known as villi as plural, and a single projection is known as villus. These villi are collectively increasing the surface area of the inner wall of small intestine in order to increase the absorption. When we discuss the detailed structure of a villus, we come to know the cells that are lining the villus, they have got their membranes projected into finger-like outgrowths. And these finger-like outgrowths of the cell membranes of the cells that are making the villus, these are known as microvilli. So what is the difference between villus and microvillus? Or villi or microvilli? Villi are the finger-like projections of the inner wall of the small intestine. Villi are the finger-like outgrowths or projections of the inner epithelium of the small intestine. Whereas the cells that are making the inner epithelium of small intestine, they also have one of their membranes facing the cavity of small intestine projected into finger-like outgrowths. So finger-like outgrowths of the cell membranes of these cells, these are known as microvilli, and the finger-like outgrowths of the tissue, that is the inner epithelium, this is known as villi. Both these villi and microvilli increase the surface area for absorption. Now, when we look into the villus, what we see, we have got a transport network in the villus. And this transport network includes centrally located lacteal and surrounded by capillaries network. So water, minerals, amino acids and glucose, when they are absorbed by the cells, they are transported into the capillaries, into the blood present in the capillaries, and later they are carried to the liver through hepatic portal vein. As fatty acids are water insoluble, so they are given to the lacteals after absorption, and through lacteals they are transported. Within small intestine, it's not only the digested food is absorbed, but water and maximum water reabsorption is done. Water absorption is done in the small intestine along with some minerals, particularly sodium ions, and some vitamins are also absorbed in the small intestine. And rest of the food that is not absorbed is known as residue. Why the rest of the food that is not absorbed here? Because that food is not in the form that could be digested, or we do not produce any enzyme to digest that food. For example, cellulose fibers that are present in the plant products. We do not produce cellulase enzyme that could digest that cellulose fiber. So that remains in the residue. Remember one thing, digestion process completes in the small intestine, and absorption of the digested substances also completes in the small intestine. If any of the molecule that remains unabsorbed, that is pushed forward into the large intestine, because during the process of digestion, peristaltic waves also continue to occur throughout the length of small intestine. So gradually, the moment food is being processed, food is being digested, it is also being pushed in the forward direction towards the large intestine. So each peristaltic wave pushes a little part of the food in the forward direction, and ultimately that reaches the large intestine. Before going to discuss about large intestine, I must tell you about liver. All the digested substances, they are carried with the help of hepatic portal vein to the liver, where liver decides the concentrations to be flowing within the bloodstream. That we will discuss later when we will discuss the role of liver in the human body. Now continue with the residue that has been pushed forward into the large intestine. Large intestine has got two segments, colon and cecum. In large intestine, there is no food digestion and there is no absorption of the digested food. That's why large intestine's inner wall is smooth. It is not projected into villi or microvilli, just like in small intestine. Its inner wall is smooth, and that is only meant for the absorption of water and minerals from the residue. At the end of large intestine, residue reaches the rectum, and it contains only the materials that are unabsorbable, that cannot be absorbed at all. So these materials are now taken as feces. They are temporarily stored in the rectum, and when rectum is filled up with the feces, so muscular contraction in the rectum wall will make them expelled out of the anus, what we call egestion. So egestion is the last process of this digestive system, where undigested food residue is pushed out of the body, expelled out of the body, out of the digestive system. One must not misconcept this egestion with excretion. Excretion is the removal of metabolic waste from the body, whereas egestion is the removal of undigested food residues out of the body, out of the digestive system. Now we come back to the liver, and we need to discuss the role of liver, that what particular role does liver perform within the body of the human. Liver is a very vital organ that performs a number of jobs simultaneously. One thing, in sport of digestion, what it does? It produces bile. Bile is extracted from bilirubin that is produced during red blood cells hemoglobin metabolism, and this bile is temporarily stored in the gallbladder, and when it is needed, this bile is introduced in the small intestine, in the duodenum part of the small intestine, in support of digestion, in support of activating enzymes, in support of maintaining alkaline pH in the small intestine. Similarly, this liver decides the concentration of glucose and the fate of amino acids that need to travel the bloodstream. In a healthy person, the blood sugar level must be in the range 80 to 120 milligram per deciliter. And this blood sugar level must be maintained. It must not increase. But through the digested food, lot of glucose enters the bloodstream. So liver takes up the excess glucose in response to the hormone insulin and converts it into glycogen and stores it. So that blood glucose concentration must be kept in the normal range, in the optimum range. Similarly, when blood glucose concentration starts declining, so at the same time, pancreas produces a hormone known as glucagon that interacts with the liver cells, and the liver, in response to that glucagon, digests, hydrolyzes stored glycogen into glucose and releases into bloodstream. So this is another job of the liver, that it helps maintaining glucose concentration in the blood. When blood glucose concentration is high, more than the optimum range, so liver takes up the excess glucose, converts it into glycogen and stores it. And when blood glucose concentration drops below normal, so this liver hydrolyzes the stored glycogen and releases into bloodstream in order to restore the blood glucose concentration and help the body cells to continuously respire. At the same time, this liver also oxidizes the lactic acid. The lactic acid produced during anaerobic respiration in our muscles, that lactic acid is carried to the liver, where liver cells oxidize this lactic acid and reduce the possible toxicity chances. Similarly, the ones who keep drinking alcohol, alcoholics. So alcohol, that is the ethanol, is also absorbed into the bloodstream through small intestine, and through hepatic portal vein it is carried to the liver, and liver oxidizes that alcohol to kill its toxic effects. So alcohol toxicity is also reduced, also minimized by the role of liver. At the same time, certain drugs that could be the part of the medicine we normally take to cure certain illnesses, or certain other drugs that could be taken as a result of addiction. So these drugs are also detoxified by the liver. So liver is playing multiple jobs simultaneously, means it is supporting the digestive process by producing and introducing bile into small intestine. It is helping in maintaining the glucose concentration with the help of hormones, insulin and glucagon. It also carries a process of oxidizing alcohol and lactic acid. It also detoxifies drugs, and urea is also produced within the liver during the process of deamination. Unwanted amino acids, they are broken down during deamination process, resulting the formation of ammonia and keto acids. Ammonia is highly toxic and it needs a lot of water to expel it, to dilute it, to reduce its toxicity. As we do not have that plenty of water, so ammonia is immediately reacted with carbon dioxide to form urea. Urea is less toxic than ammonia, can be retained in the body for some time, and it needs less water to be excreted. So urea is produced by the liver during deamination process and is later carried to the kidney, where it is filtered out and excreted out of the body as a component of urine. Whereas keto acids, these are important organic components. If there is an immediate need of glucose, keto acids are immediately converted to glucose and that glucose is respired. But if there is no immediate need of glucose, these keto acids are converted into glycogen, and when needed, that stored glycogen can be converted to glucose and used. But if liver is already having pre-stored glycogen, as a healthy adult liver can store around 100 gram of glycogen, and it is already pre-saturated with the glycogen, so no more glycogen can be stored. So these keto acids are converted into fats. Some of the fats are stored in the liver and some of the fats are transported to the adipose tissues, where they are stored in the fat cells. So this is the job of the liver. This is another job of the liver. So in this human nutrition, we have discussed the role of small intestine in the process of digestion and absorption. We have also discussed the role of liver and pancreas in support of digestive activity in small intestine. We have discussed the role of large intestine, and we have also discussed the multiple roles performed by the liver. And remember, assimilation is actually the utility of these absorbed substances into the bloodstream, just like we have discussed about liver, that it takes up glucose and converts into glycogen. Similarly, body cells, they take up glucose for their respiratory needs, they take up amino acids to make their required proteins, and mineral ions for their needs, and fat molecules, fatty acid, they take up to make their membranes. So this is what we call assimilation. So with this discussion, we have ended up with the episode of human nutrition, and with this we have also completed the human nutrition discussion as well. So stay tuned for upcoming topics. Thank you very much.